Once post-translational modification has finished, the proteins are grouped into different taxi vesicles depending on which wristband modifications they received earlier. Now Patrick and his protein pals have left the trans-Golgi network. They either travel to the lysosome or are excreted from the cell via regulatory or constitutive pathways. In the regulatory pathway, proteins are grouped together into concentrated vesicles. These vesicles are pulled along microtubules by motor proteins. Once the vesicles reach their destination at the phospholipid bilayer, they are held up by a red light. They must wait for the correct signal before they can exit the cell. The signal to exit the cell could be an action potential, hormone, or other external signal. Once the vesicle receives a signal, it gets a green light to leave the cell and carry on its journey.
Constitutive secretion occurs in all eukaryotic cells. Soluble proteins are usually secreted by this pathway. It involves the passive, bulk flow mechanisms of proteins out of the cell. If no modifications are made in the Golgi, the proteins will enter the default secretory pathway and are packaged into vesicles and sent directly to the plasma membrane for immediate constitutive secretion. The vesicles have a clathrin coat, and the vesicles are used to maintain the proteins and lipids in the plasma membrane by replacing them with newly synthesized ones from the Golgi. A Rab8 protein is used to specifically target the vesicle to the membrane and dock it there. Patrick's friend Polly decides she's had enough of waiting around at the Golgi, and by this point is totally phosphorylated. She hollers for a secretory vesicle to pick her up and take her to the outskirts of town, the phospholipid bilayer. The taxi driver uses his SAT-Rab8 for direction to the membrane, where it will be recognized by a snare protein. The vesicle uses GTP and tethering proteins to tether the vesicle to the membrane before the snare proteins twist to allow fusion of the membranes and exocytosis. Unlike the regulatory pathway, the constitutive pathway is independent of hormonal or neuronal stimuli and does not have a significant role in protein concentration or storage. Like before, pause here to review the processes of regulatory and constitutive secretion.
Created using Powtoon.